Okay, let's discuss about ribosomes. So ribosomes, we have already discussed some of the important features in the first, second class. So ribosomes, are these membrane bound or non-membrane bound? What do you think? Are these membrane bound organelles or non-membrane bound?
Non-membrane bound.
Yeah. So these are non-membrane bound cell organelles. They are not surrounded by any membrane. So ribosomes are very, very small structures. We usually use the term particles. So these are dense particles, and these can be observed only with the help of an electron microscope. The structure of ribosomes can be seen only with the help of an electron microscope because of their very small size. And these were first observed by a scientist named George Palade in 1953. So that is why ribosomes are sometimes referred to as Palade particles. So they are called Palade particles because these were first observed under the electron microscope by a scientist named George Palade. Now, what are ribosomes made up of? Ribosomes are mainly made up of two things. First one is RNA, that is ribonucleic acid, and second one is proteins. So ribosomes usually have two subunits. There is a large subunit like this, and there is a small subunit. Okay, so this is called the large subunit, and this is called the small subunit. And these ribosomes are composed of both RNA and proteins. RNA and proteins. But the type of ribosomes which are found in eukaryotes and in prokaryotes, these are different. So something we have discussed in the first class was that ribosome is a cell organelle which is common to all the cells, whether it is a bacterial or prokaryotic cell, plant cell, animal cell, fungal cell, almost all the cells will have ribosomes. But the type of ribosome which is found in eukaryotes and the type which is found in prokaryotes, these are different. So in eukaryotic organisms, the ribosomes will be 80S, and prokaryotic organisms, the ribosomes will be 70S. So S here, it actually stands for something called sedimentation coefficient or Svedberg's unit. It is called sedimentation coefficient or Svedberg's unit. So this is actually a measure of both density and size of the components. Okay. So sedimentation coefficient or Svedberg's unit will be a measure of density and size of these particles. So let's see 80S ribosome. So 80S ribosome is found in eukaryotic cells. So it should also have a larger subunit and it should have a smaller subunit. Similarly, in prokaryotes also there is 70S. S refers to sedimentation coefficient. It should also have a larger subunit and a smaller subunit. Now let's see which are the subunits found in 80S. So 80S is made up of 60S, that is a larger subunit, and smaller subunit is 40S. In 70S ribosome, this larger subunit is 50S and the smaller subunit is 30S. So if you look here, if you try to add these values 60 plus 40, it should be 100 and 50 plus 30 should be 80. But when these ribosomes, so that is a separate, when these subunits are taken separately and when their density or size is measured, then you will get 60S and 40S. But when these two combine together to form a single unit, that is one large ribosome, then their density will kind of change or decrease. It will be only around 80S. Okay, so 60 plus 40 is going to be 80 here. 50 plus 30 is going to be 70 here. So you should not get confused between what are the subunits. So always remember that 50 plus 30 is going to be 70S, not 80S. And 80S is found only in eukaryotic cells. 70S is found in prokaryotic cells and two cell organelles. One is mitochondria, and it is also found in plastids, that is chloroplast. So both mitochondria and plastids are cell organelles containing 70s ribosomes. So these are the important things that you should remember with respect to ribosomes. Ribosomes are dense particles. They are very, very small in size. First observed by George Palade. That is why it is also called Palade particles. It is composed of two things, RNA and proteins. It is non-membrane bound. There are two types of ribosomes. 80s is found in eukaryotes, 70s is found in prokaryotes. Each ribosome is made up of two units, a larger subunit and a smaller subunit. In eukaryotes, the largest subunit is 60s, smaller subunit is 40s. In prokaryotes, larger subunit is 50 and the smaller subunit is 30s.